My name is David Holden, and I'm currently the Regius Professor of Infectious Disease at Imperial College London. And I work in a centre which is devoted to the study of bacterial physiology, pathogenesis, etc. So the centre actually is rather unique in that it's, I think, the only centre in the world with an exclusive focus on bacteria. So that's what gives it its identity. My lecture today really reflects the work that I've done over the last 40 years by focusing on 3 genetic screens. One of which I did very early on in my career and wasn't very important, but something about that screen stayed in the back of my head and became fundamental in thinking about the second genetic screen, which is basically this barcoding signature tagged mutagenesis method that I invented and we developed as a group in the mid-90s. And that, what we learned from that, the biology that came from that, provided the basis for a third genetic screen, which we did in mammalian cells, to enable us to understand the function of one of the proteins that we identified as a result of the second screen. So I hope that those 3 screens which are linked provide a kind of basis or framework for the talk that I'm going to give. What is STM? So that's signature tagged mutagenesis, a rather clumsy title, but it's essentially barcoding mutants. So if you want to understand the function of a gene, the very common and traditional method has been to mutate it, to inactivate it in some way, and then ask whether the cell or the organism that contains that mutated gene has a different property, a different phenotype. And so when you're studying pathogenic bacteria like Salmonella, which may have a genome of 4,500 to 5,000 genes, and you want to determine the function or find the genes that are required for growth in a host, that becomes a very laborious and expensive and ultimately ethically challenging enterprise to do, because you would need to inactivate genes one by one and then examine their virulence in a model of infection, in our case, a mouse. So this becomes very difficult to do. So the concept behind STM, or mutant barcoding, is to do the mutagenesis of the organism, but for every mutant to integrate into that mutant a barcode, a unique sequence identifier. And that's what we did. And that then enabled many mutants to be combined together in a kind of multiplexed manner so that we could examine them en masse for their virulence properties in an experimental model of infection together, and allow the organism to tell us which were attenuated and which were not by comparing the barcode abundance in the mutants before and after the infection. So each barcode is unique to each mutant, and that then enables us to disentangle the mutants after an infection and understand which ones were attenuated in virulence and which ones were not. So that essentially is the principle of the method of mutant barcoding. And we applied it to Salmonella. And there we had a great stroke of luck, because we discovered this very interesting biology, which I've been studying ever since. But the STM method has subsequently been improved upon, adapted, and modernized. And so the modern iterations of the barcoding approach are much more sophisticated and impressive than that original rather clumsy method we used all those years ago. Why it was important, I think, is that it greatly simplified and speeded up the process of finding interesting genes. And not only that, sort of practical benefit of STM, the barcoding method was exciting because it was more broadly applicable. So immediately you could see applications of STM and its derivatives across a range of different biological systems. So we used it for Salmonella, but it's been applied to study virulence in most other pathogenic bacteria, but also pathogenic fungi, parasites, and in human cells. So the application of this technique then gives rise to understanding biology, and then that understanding of biology can be used ultimately to not only understand, but to be used practically either in vaccine design or in treatment or in ways that I think are just unpredictable at the moment. But the more biology you understand, the more you have at your disposal to exploit in the future. Having applied the technique to Salmonella and discovered this SPY2 injectosome, we and others showed that it's important for Salmonella to grow inside host cells and to suppress immune function, both innate and adaptive immune activities. Now, a lot of people have thought, well, that looks like an interesting target that you could maybe exploit in therapeutics. But I personally don't think that targeting something like the SPY2 injectosome is necessarily the best way to exploit this knowledge, because if you inhibit the injectosome, you'll stop the bacteria from growing, but you won't necessarily kill them. And obviously, to treat infections, you ultimately want to kill the bacteria. But I think the applications of the biology that we and others have discovered about SPY2 and its effectors are more likely to come in areas such as vaccinology, in cancer treatment, and in ways that are simply unpredictable at the moment. What's clear is that these effectors, not only from Salmonella, but for many other pathogenic bacteria, have got absolutely fascinating and exquisite biochemical functions which are not necessarily present in eukaryotic cells. And they therefore give you a toolbox of biochemistry to exploit. And I don't think it's possible to predict at this stage, the many ways in which that exploitation is going to happen. We just have to wait and see. In the context of this prize, I'm obviously very grateful, but there are a huge number of people who I should really thank for their contributions over the years. And I've been extraordinarily lucky with a whole range, right from the very beginning of my career, of talented and motivated and fun people to work with. And to a great extent, they deserve the credit and part of the pleasure of this prize.
I'm delighted to welcome Professor David Holden of Imperial College to deliver the Prize Medal Lecture for 2024. The Prize Medal is awarded to an outstanding microbiologist who is a global leader in their field and whose work has had a far-reaching impact beyond the discipline of microbiology. I've known David for many years, and I can say safely, in this case, we have an outstanding recipient. He is best known for inventing signature-tagged mutagenesis, and its derivatives have been applied to numerous bacterial and fungal pathogens, parasites, yeast, and mammalian cells, whereas usually combined with CRISPR-mediated mutagenesis. Collectively, these studies have identified many thousands of interesting mutants, large numbers of which have been followed up to provide mechanistic insights into different biological processes. It is my honor to introduce David, whose prize lecture is From Barcodes to the Biology of Salmonella. David.
Thank you very much, Del, for that introduction. It is a great honor to receive this award, and I'm very grateful to the society, the officers, the committee who decided on me for this award, and also my mysterious supporters, nominators, not looking at anyone in particular, but thank you. Actually, this is a bit of a homecoming for me because I went to school in Edinburgh, just across the canal near Brunsfield. And I was walking around there yesterday and bringing back lots of memories of my youth. And also, I gave my first talk as a PhD student in Edinburgh at a microbiology society meeting back in 1980 something. And I've been a member of the society for most of my professional career, so it means a lot to me. It means a great deal to me to receive this, and thank you. So what I'm going to do today is, as Del intimated, talk about our barcoding technology and how it came about. And this really involves the description of 2 genetic screens. And then I'm going to finish my talk by talking about some biology, very interesting biology of Salmonella, which involved a 3rd genetic screen, which is a sort of inverse of the one that we used on Salmonella to find the genes and the proteins that were involved in the process that led to this 3rd screen. So they're linked in a way. So it's a tale of 3 genetic screens, but before I go any further, I also have to say thank you, a huge thank you and acknowledgement to all the people in my group who've contributed, more than contributed, actually done the work over the last 30-odd years or so. Right from the very beginning of my time as a lecturer, I've had extraordinary good luck in working together with young people who've been inspirational and role models and a whole lot of fun as well. So obviously I could tell stories about each of these people, which would take up most of the day, but all I can do is show their names and express how grateful I am to them. So the story for today begins when I was a postdoc in North London at the National Institute for Medical Research. And I came there having done a postdoc in the US, working on a fungus called Ustilago maydis, a pathogen of maize. And I came to work at Mill Hill because of this fellow, Robin Holliday. Robin had used Ustilago as a model system to come up with his beautiful 4-way Holliday junction structure, which we now know to be the central intermediate which enables recombination of DNA in all domains of life. And Robin was a kind of a hero, really, and I was thrilled to have the opportunity to come to Mill Hill and do a postdoc with Robin. But as I arrived at Mill Hill, the day I arrived, I was walking up the steps to this grand institution, and Robin was coming the other way. And he stopped on the steps, and I said, "Robin, I'm David Holden. I've come to do a postdoc with you." And he went, I have to tell you that I'm emigrating, actually, leaving today. But don't worry, you'll be in good hands, and off you go, and have a great time." So that was my sole, pretty much my sole interaction with the great man, but it didn't stop me working on Ustilago. So Robin, this central intermediate structure involved the product of a gene, and Robin had come up with all of this through classical genetic analysis. He was a visionary, a true visionary. You could see the molecular detail from classic genetic analysis. And central to this was this gene, the REC1 of Ustilago. So I had in my time in the States, become a gene jockey and learned at the cutting edge how to do cloning, recombination, and transformation of Ustilago. And so I set about trying to clone this gene by incorporating into plasmids large DNA segments from wild-type Ustilago and then transforming them into a REC1 mutant. And the critical thing about a REC1 mutant is its hypersensitivity to UV light. So this provided a very convenient selection. Obviously, if a plasmid, a cosmid, contained the functional gene, then upon transformation, we'd expect the recipient to become UV resistant. So we did this in pools. So this is positive selection from pools. And we made many of these, transformed them, or I did, transformed them into the REC1 mutant, subjected them to UV light, left them for a few days, and then I got one colony after several thousand transformations, I think. And this turned out to contain the REC1 gene from one pool, which we then went on to do individual transformations to identify that single clone containing the REC1 gene, and then we could begin to characterize it, which we subsequently did. But that's not the point of this story. What I want to emphasize is the fact that this screen involved gain of function and positive selection from pools of mutants. So I didn't really think about this very much at the time. It was very straightforward. None of us would pay any attention to this now. And I think it possibly lodged in the back of my mind the idea of using pools and thereby exploiting my inherent laziness. And there it was left. I didn't do much more work on recombination. Instead, I took the opportunity to go to the Hammersmith Hospital in London to work on a fungus that I knew nothing about, but was causing problems in bone marrow transplant units and other environments where people were immunosuppressed. And this pesky airborne fungal spore, Aspergillus fumigatus, would get in and cause very serious life-threatening infections. But hardly any molecular genetics had been done on this fungus. So I took the opportunity to start working on it. And I read the literature and tried to look for clues as to what sort of proteins or toxins the fungus might make that would account for its ability to cause disease in the lung. And reading the literature, we came across this alkaline protease. And so using my molecular genetics, we set up a transformation system for Aspergillus. And with PhD student Christoph Tang, we knocked the gene out, and then we tested the virulence of this mutant in respiratory infection models in mice and in rabbits. But this mutant turned out to have no detectable virulence defect. And so the word "not" features in the title and was to subsequently become quite a frequent word over the next 3 or 4 papers, where we made more and more mutants on what we thought were plausible virulence determinants, like this restrictocin, which is a potent toxin, and made double mutants. But again, you can see that we were unable to see significant differences between the mutant and the wild type. So this all involved a lot of work and a lot of time, and the negative results began to get to me. I began to feel a little bit frustrated, a little bit depressed, having to go to conferences and present negative data, and completely aware that we needed a different approach, that this sort of guessing game was finished. I wasn't going to do any more of that. I'd rather leave science than continue trying to cherry pick what I thought might be important. Too risky. And at this conference, I went to a poster session. And I started looking at a poster which had nothing to do with virulence or anything like that. This was a poster describing a way of mapping chunks of DNA along the chromosome of a fungus. And it was called chromosome walking. It's been superseded now, of course. Nobody uses techniques like this anymore, I think. But it was interesting because what was described was the cloning of large chunks of fungal DNA into cosmids, put onto filters in E. coli, and then one of these would be chosen, the DNA extracted, labeled, and then hybridized back to colonies on the colony filter. And so you'd expect to get, obviously, the one from which you took the DNA would hybridize, but also if there was overlapping DNA, another colony might light up. But the key thing was that the next point in the exercise was to take DNA from a clone that hadn't hybridized. So you take it without sampling, without replacement. Label that one, hybridize back. You get new colonies, but you also get overlapping colonies, and that tells you that those two segments of DNA are overlapping. And then along this kind of process, you can build up a map of the chromosome. But that didn't really have any interest to me. I wasn't interested in chromosome walking. I was just conscious of the fact that he was looking at negatives, that the negatives were of interest. And again, that registered in my mind because when we're looking at pathogens and we want to make mutants to find genes that are important for growth, it's the negative, the one that fails to grow, that's of interest. So that registered with me, and somehow on the way back on the aeroplane back to my home in London, I was thinking, I couldn't stop thinking about this. I was thinking about pools of mutants, the fact that we were interested in the negatives, in loss of function, and the use of hybridization. And that evening, I continued to think about it, and slowly began to emerge an idea that if we could make mutants and actually barcode them in some way, give them identifiers, that would then enable us possibly to pool them together and track them, allowing the mutants to be screened en masse. And then there would be, if we made random mutations, there would be no guessing. There would be no, this looks like an interesting candidate. We would let the organism do the job for us. And so I came up with this barcode design, which basically involves synthesizing a stretch of DNA, which is characterized by a central region of 40 nucleotides or so, which is hypervariable. So we just asked the synthesizer to put in any of these nucleotides in these positions. And this would generate so much variability that upon cloning, the chances of getting the same sequence twice is next to nothing. And these variable regions, which were enriched in Gs and Cs, which could be selectively radiolabeled, were flanked by arms of invariant sequence. And this would enable all of the signature tags or sequence tags to be co-amplified, but then the arms could be released with these restriction enzymes to reveal these selectively labeled central regions, which would consist of unique signature tags. So to explain this a little bit further, this is the outline of the procedure that we came up with. So here you have these signature tags that I've just described, with the central region being prone to selective labeling, flanked by invariant sequences. They could be co-amplified together, and then cloned into transposons to be their vehicle for mutagenesis. So that basically transposon mutagenesis into fungi or bacteria, well, bacteria in this case, and each transposon would carry a unique tag. But the flanking arms would allow them to be co-amplified, labeled, and used as probes. So the mutant bacteria were assembled into pools of 96 on 96 well plates. They were pooled together and then inoculated into mice. And we chose Salmonella, not because I wanted to start working on Salmonella. I hardly knew anything about it. I knew it was a bacterium, but was it a gram-positive, a I honestly had very little idea. But I did know that there was a good model of infection in mice that recapitulated to some degree features of typhoid fever. So we used this model just really as proof of principle. I always planned to go back to work on fungi. And so we injected these mice with the input pool, waited a few days, and then from the spleens of these animals, recovered bacteria, plated the spleen homogenates onto agar, bacterial colonies grew up. We PCR'd up the tags from bacteria which had grown, and then compared the input and the output pools on filters. That was the idea. So screen 2 that I'm talking about today took 12 months, basically, from concept to test. And this is an early result that we got in the lab. So here's the input pool, where 96 Salmonella typhimurium mutants each carry a distinct barcode. So these don't cross-hybridize with each other. We did, obviously, these controls to ensure that they were specific probes for their mutants. And then this is an example of pools recovered from different mice. So here's an output pool, and you can see that there are 3 mutants which don't come up on the output pool. So these are attenuated in virulence. This would be predicted because alongside genes that have been selected for by the mammalian environment and immune system, there would be other mutants that were auxotrophs, for example, which were already known to obviously be attenuated for growth. So finding mutants that were attenuated were not surprising, but the most important control was really to do the same pool in a second mouse and make sure that these were not sort of stochastic negatives and that they were reliably selected against. And this was an extremely exciting day for me. All that negativity, all that depression about our work on fumigatus was suddenly worth it when the technique was shown to work. And here you can see the same 3 mutants missing from the second animal. So this isn't stochastic, it's real. These mutants are attenuated. And also, the thing to say, which probably maybe doesn't apply now because we have the genome sequences for everything, is that it was so exciting to not to know immediately what the gene was, right? So there was a period of time of expectancy when we were waiting for the results of the sequencing to say, what had we hit? And the genome sequence wasn't known back then, so this was really, really exciting, and some sleepless nights there until we got the sequences back from sequencing off the ends of the transposons. And here you can see that a whole cluster of these different mutants, these lollipops, denote the insertions of the transposons in what we call now Salmonella pathogenicity island 2. And the first thing that was apparent was that some of these genes were similar to those encoding a type 3 secretion system, but not the type 3 secretion system of the flagellum, the type 3 secretion system that had already just been described in Salmonella and in Yersinia and other bacteria as being virulence-associated type 3 secretion systems, what we now call injectosomes. So these were similar to an injectosome, and one, which was encoded by Salmonella pathogenicity island 1, had already been described in Salmonella and shown to be important for the invasion of non-phagocytic cells by Salmonella. So this immediately told us that there was a second type 3 secretion system in Salmonella, but this one was not involved in invasion, but for survival in the systemic phase of the disease. And so we wrote the paper up very confidently and sent it to Nature, where it was promptly rejected without review. Thank you very much, no thanks. So, but we persevered and we sent it to Science, and it was published in 1995. And I want to acknowledge two very important people who are behind this work, Michael Hensel and Jackie Shea, who decided with me to stop working on Aspergillus for a time at least and see if we could get this method to work in Salmonella. And it worked. And so we were also very lucky. We were very, very lucky that there was this kind of biological pot of gold there in the Salmonella genome that we could then work on the biology of. So this barcoding technique, as Del mentioned, it's basically the conceptual basis for all multiplex mutant screens that involve generating mutants with barcodes. So now you can use, you can use synthetic barcodes or guide sequences, mixing them to create an input pool, subjecting that pool to selection, collecting an output pool, and then comparing the barcode abundance in these different pools to identify mutants of interest. And that approach has been applied now, as Del said, in many, many different systems, in bacteria with derivatives of STM like TNSEC, etc. But there's an important lesson for young people here, having gone through this whole thing, which is if you're doing your PhD or one of your first postdocs and it's not going well, don't worry. Failure is very important. It's intrinsic and essential, and I know for sure that if I didn't have those years of difficulty, I wouldn't have come up with this idea. So I think the crucial thing is just relentless curiosity. So going now from the genetics to biology, what does this thing do? That was always the plan, to work on interesting biology. So many groups have subsequently contributed to our understanding of this type 3 secretion system and their effectors. So these effectors are actually translocated from the bacterial cell into the cytoplasm and into membranes of the host cell, whether they be epithelial, macrophage, or of dendritic origin. So it's basically the acidification of the vacuole and nutrient deprivation that leads to the assembly of the injectosome. And then a very great colleague and postdoc in the lab, Susan Yu, showed that the delivery is exquisitely controlled. And by sequential sensing of the low pH of the vacuole, and then when this pore is formed in the vacuole membrane, the sensing of the neutral pH of the host cytoplasm to enable selective delivery of effectors into the host cell. So what do these effectors do? So again, many groups have contributed to our understanding of the functions of these effectors, and their physiological functions break down broadly into 3 or 4 groups. The first is that several of these effectors, circled in red, are involved in controlling the vacuole membrane. So for the most part, Salmonella replicates in cells inside a vacuole, and the vacuole protects it, provides a niche, and it's a kind of detoxified lysosome, if you like, a late endosomal compartment. And it devotes at least 6 of these effectors to controlling the expansion of the membrane, the composition of the membrane, the scission of the membrane as the bacteria divide, etc. But other effectors are involved in organelle tethering in epithelial cells that hold these Salmonella vacuoles close to the Golgi apparatus, where they probably gain nutrients from interfering with secretory traffic. And then several other effectors are involved in inhibiting innate immune signaling. And many bacterial pathogens use their type 3 secretion systems to block or evade innate signaling mechanisms. But it's known that Salmonella faces an adaptive immune response in long-term and chronic infections. And there's this one effector that we know about, STD, which I'm going to spend the rest of the talk discussing, which is responsible for the interference of T-cell responses. So these CD4 T-cell responses are crucial for the control and elimination of Salmonella in mice and in humans. So there are very good GWAS studies showing that MHC2 controlled CD4 T-cell responses have a huge effect on your ability to control and eliminate Salmonella, and similar studies in mice. So the question arises whether there's been enough time in evolution, the association of Salmonella with the mammalian host, in the order of maybe 150 million years or so, for that to be enough selective pressure to force or select for the emergence of a protein that can interfere with that response. So very briefly, antigen is expressed on antigen-presenting cells via MHC class II molecules to CD4 T cells, and these T cells promote a potent cytotoxic response. But these molecules, these MHC2 molecules, are first transported to the plasma membrane bound to an invariant chain, and they're then internalized back into the host cell, into the MHC2 compartment, where the invariant chain is swapped for antigen, which is loaded onto the antigen loading site, and then transported back to the plasma membrane for recognition by the TCR. So that begs the question, do any of these SPY2 effectors suppress T-cell responses? Well, it was already known from quite a few studies, here's a couple of examples, that Salmonella seems to be able to inhibit surface class II expression on dendritic cells, and that this process seemed to involve ubiquitination of surface MHC2. But the mechanism, the effector responsible and the mechanism were not understood. So we tested, by this time we'd got maybe 30 or so effectors identified, and so we had individual mutants in all of these effectors, and we simply tested them for surface class II in infected antigen-presenting cells. And here are the results. So here's the surface MHC2, and you can see the wild type is low compared to the SSAV, which is the SPY2 null. And most of the mutants behave like the wild type, but this one is back up to the SPY2 null. And this is STED. So STED is only 111 amino acids. It's a small protein. Thankfully, it is not only required, but it's sufficient to reduce surface peptide-loaded MHC class I. So it doesn't need any other help from Salmonella proteins, which makes it convenient to study. But also, it's an integral membrane protein. It's very small, so on the cytoplasmic face, there are not a lot of information available to mediate that very dramatic effect. And by the way, the effect is very specific. This protein doesn't touch class I, and it doesn't touch the invariant chain-loaded class II, only the peptide-loaded. So it's very specific. So how does it work? So this led to screen 3, because the hypothesis was that there wasn't enough information. It didn't look like an enzyme. So the most simplest explanation was that it might use some host proteins to mediate its effect on class II. Maybe it would co-op or hijack something in the host cell to have its effect on class II. So this led to my third genetic screen, and this one I really like, because aficionados of these screens will know that it's a very powerful one if you have genetic loss of function for physiological gain of function. So we did a CRISPR-Cas9 screen, pools of tagged mutants, this time guide RNA, input, screen by input and output, not too dissimilar to what I've already described, but this time for human proteins, which are required for STD-mediated surface depletion of MHC class II. And so here's the outline of how the screen worked. So in these antigen-presenting cells, they're naturally high in class II, this red on the cell surface. Transduce them with GFP-STED, they're fluorescent, and the antigen-presenting protein is gone. So then we make mutants by CRISPR mutagenesis and sort, using FACS, for mutants where STED has lost its ability to suppress class II on the cell surface. And then, after 3 rounds of sorting, subject them to sequencing. Here's an example of microscopy of STED in cells where there's no MHC2 on the cell surface anymore. It's all internalized. But after 3 rounds of cell sorting, there's a massive amount of class II on the cell surface. So the screen worked, but were there interesting genes that we found? Yes, we found 2. The first is called TMEM127, and very little was known about that protein, except that it's a tumor suppressor. And mutations in this gene give rise to a rare carcinomas. Whereas WWP2, which was the other hit, a lot was known about it. It's one of a family of so-called NED4 E3 ubiquitin ligases. And interestingly, it's also, when dysregulated, an oncoprotein. So they're both involved in cancer, but in different contexts. But a lot more was known about this ubiquitin ligase, which basically takes a ubiquitin molecule and deposits it on a substrate. And what we found, I will spare you the experimental detail, but what we found, this is all published, is that TMEM127 is an adapter for WWP2. And TMEM127 has this PY, this proline tyrosine motif, and that is required for its ability to interact with WWP2. And so, as a result of that, finding that, we can say that 127 is a new NED4 interacting protein, or ND-FIP. So E3 ligase is a NED4 family E3 ligase, and TMEM is an ND-FIP, a NED4 interacting protein, of which a few had been described. We also found that TMEM binds to STED. Interesting for that reason, because it was discovered in the hit for STED function. And we then found that STED actually bridges this complex of the adapter and the ligase to MHC2. So that's depicted here. So without STED, TMEM, it's a 4-membrane spanning domain protein. It's got this PY motif at the C terminus. That we showed is required for binding to WWP2. And STED, when it's present, brings this complex together with MHC2, and then WWP2 can start to ubiquitinate MHC2 and lead to its internalization and degradation in the lysosome. But that's really not very satisfactory. That just tells you this binds to that, that binds to this. What's the actual mechanism? Well, this is where we are at this point. So TMEM127 can bind WWP2, but we don't know anything else about this in the absence of Salmonella, just that these two combine. Then STD is produced. It takes a rather circuitous route to the MHC2 compartment via the Golgi, where it picks up a pathway involving a protein called AP1, which takes it to the MHC2 compartment, where it then can engage its targets and ubiquitinate them, degrade them, and reduce surface MHC2. Because this surface molecule is constantly recycling between the antigen loading compartment and the cell surface. So if you start degrading this, eventually the surface molecule is removed. But what is the mechanism? That really wasn't a completely satisfactory explanation. So, in the last few minutes, I just want to, have I got time? 10 minutes. Oh, that's good. Okay. So the catalytic activity of WWP2 is normally very tightly regulated by autoinhibition. So they're normally folded, it's folded upon itself and inactive. And it needs to be activated by these ND-FIPs. And a good example of this in the host cell is this ND-FIP2. And this allosterically activates WWP2. And once the protein is opened up, it can begin to function. And conveniently for us, the activation of the E3 ligase can be detected in an assay by the fact that it becomes auto-ubiquitinated. So that's shown here. So normally WWP2 is held in this folded inactive state. NDFIP2, not the one that we found. This is an endogenous protein known to activate WWP2. It's got 3 PY motifs, and these interact with these central protein-protein interacting domains of WWP2, and this helps to open up the complex and expose the HEC domain, which is the catalytic domain of the E3 ligase. This can bind ubiquitin and also transfer ubiquitin to a substrate. So that provides a convenient assay to start looking at the activation of WWP2 by TMEM127. So and this is the only, I've got 2 blots, that's all to show, so don't worry. So this is the assay. We transfect mammalian cells with proteins of interest and with His ubiquitin, His-tagged ubiquitin. Don't ask me why the assay is done in this way. It's just what's done in the field, and it works. And then after they've been allowed to interact and work, if they do work with each other, are subjected to His pull-down and then immunoblotting. So in this first example, there's no STED. This is just the positive control to show you that in the presence of WWP2 and NDFIP2, so this is the positive control. We already know this activates WWP2, and in the His ubiquitin pull-down, you can see here activated, ubiquitinated WWP2. And that's completely dependent on this PY motif, or these PY motifs, 3 mutants in NDFIP2. If they're substituted for alanines or something, then there is no ubiquitination. But TMEM127, if that's added instead of NDFIP2, wild-type TMEM127, the one that we identified with WWP2, there's no activation. So TMEM127 by itself does not activate WWP2. So that's basically just illustrated here, whereas the bona fide NDFIP does. So that raises the question of whether STED itself has any role in the activation. And so when we add STED into the mix and together with either wild-type WWP2 or a point mutant of WWP2, a catalytically dead mutant of WWP2, suddenly, with TMEM127, not the bona fide NDFIP, we now get activation of WWP2, which of course is dependent on the activity of the protein, the ligase that we added. But that activity is not enough. You also need TMEM127. So in cells which lack TMEM127, there's barely any stimulation of activity of WWP2 by STED. So that leads to the conclusion that this little protein, which has, if you saw from the depiction, only about 60 amino acids at the N terminus and 11 at the C terminus in the cytoplasm, has got the remarkable property of binding both to MHC2, its target, and to TMEM127 in the membrane, by the way, and furthermore is not just an adapter that brings these molecules together, but has another crucial function, which is to activate the E3 ligase, because TMEM127 is itself not sufficient. So that is a novel mechanism of activation of NED4 E3 ligases, and this is very appealing to me in a broader sense, because the expectation in the field for a long, long time has been that studying bacterial effectors might not only lead to improved understanding of virulence, but also might shed light on host proteins and their functions. And so here we found a function for TMEM127. So this is the final sort of cartoon showing how we think it works. Of course, we don't have structural information at this point, but that would be the next step. But that these 2 proteins, WWP2 and TMEM127, can interact, and presumably there are other targets, endogenous targets, for this complex that we don't know about, that they are bridged by STED when it's present, that STED, in addition to bridging them, I haven't shown you the data, but it binds a N-terminal region of WWP2, and this together with the PY-mediated binding of TMEM can activate WWP2, and basically all of the proteins in the complex get ubiquitinated, even STD, and then they're degraded by K63-linked ubiquitination in the lysosome. Very finally, to say that MHC2 is not the only target of STD. It has another target, which we found by proteomics, which is called CD97. And we found that this protein is important for stabilizing the immunological synapse between antigen-presenting cells and T cells, as well as being a potent activator of T cells. So this final slide then just depicts this, that whether you have CD8 or CD4 T cell engagement in dendritic cells, if Salmonella is present, then STD can recruit these host molecules to cause the degradation of both MHC class II and CD97, depleting them from the cell surface, and then releasing prematurely the interactions between T cells and dendritic cells, and thereby dampening or inhibiting an adaptive immune response. So that's my summary and perspective, but I think I've already said most of these points hopefully clearly enough. And just to acknowledge those people in my lab who have been doing all this work over the last few years on STD, this one little protein, one of the 30. And by the way, many of these other proteins, there's lots and lots of biochemistry still and mechanisms still to understand properly. So, there's a vast amount of work still to be done. Ethelbea Santos, who found STD responsible for the phenotype in the first place. Eric Alex, who did this CRISPR screen and found these two host proteins. Camilla Godley, who was a postdoc at the time, did the cell biology on the trafficking of STD. Andrei Cherny, who found CD97 as a target for STD, now back in Prague. And Lisa Blundell, who's a medical doctor who's doing her PhD in my lab and has done this work on the activation of WWP2, together with great support from Romina Tocci and Mei Liu. I think that's it, and oh no, we don't take questions, do we? Okay.